sediment at these depths. Just thinking about, how far that sediment had to travel down the water column to settle, and thinking about sort of in your nearshore environments, it's more granular because of just it settles faster.
Is this a close enough zoom for Daniel to be able to analyze?
Suspension, yeah.
Yeah.
Oh, yeah.
Micro mollusk identification on the fly.
Easy peasy.
I mean, there's probably millions of them floating around us.
Yeah.
I know. Lots of data
Actually,
points right there.
Are we pulling a Niskin right now?
We are just waiting for...
That's a joke.
Okay.
Otherwise, it'll be another sediment core in the Niskin bottle.
Exactly. There's one way to do it. We're gonna get super creative. Let's take sediment samples with every tool we have.
Somebody was telling me, maybe it was Ben, the other navigator, that there was one expedition where they put a pair of bongo nets on top of an ROV and just drove around the bottom and sampled plankton that way.
Wait, on this ship?
No, not on this ship, on a different cruise. But
wow,
it could be an effective way of getting microfauna out of the sediment, just stirring up the bottom and just running through it.
That's a new one. Bongo nets on an ROV.
I have a visual like a butterfly net in a field of butterflies.
Exactly.
It's on top of a...
Yeah, it does seem like a not very efficient way to do mid-water sampling.
Alrighty. 10 meters left on this move. Science, do you want to stop and look along the way for these moves, or just keep our progress going towards BF?
For now, let's keep our progress going towards BF.
Roger.
Thank you.
So Paul's in the Herc seat now, as we're waiting
Paul.
for the vehicles to catch up. You good with another ship move?
Yeah, definitely.
Bridge, Nev. 30 meters, bearing 036.
Julia, I had recognized your name from somewhere, and I had to look it up, but I actually remember now, we worked together in 2010 during an Aquarius mission in the Florida Keys Sanctuary. Do you remember that?
Yes.
Yeah. Yeah, I was there as an undergrad, as working in a lab doing reef fish surveys, and I remember you were, I don't know, were you the education outreach coordinator for that also?
Yeah, I was out with, that was with Mitchell Tart and Kate Thompson and team, and we were out there trying to do some work in the Aquarius. And it was challenging because there were some issues with being able to access the Aquarius, but there was some neat reef work going on that we were, and then my job, was to basically communicate that. That was a neat expedition. I did get to dive the Aquarius on the outside. I couldn't go inside, but it was a fun dive.
Yeah, I was part of the surface dive team as well. But yeah, I remember that. It was a lot of fun.
Definitely.
And 10 years later.
World.
Yeah, 10 years. Wow.
10 years. I know. Don't say that. Time is flying by.
10 years later, you're in the deep sea. So.
And here, and now we're at
next stop
on the west coast in really cold, deep water with a lot of sediment.
Next stop, Marianas Trench.
Logical flow of things.
Yeah.
Well, it's interesting. I am going to be participating in a presentation coming up comparing the west coast to the east coast at the same latitude and off of Gray's Reef and looking at the similarities and differences, and there's going to be a lot of differences, of course, but it is interesting thinking about just how the oceanography shapes the biological communities, and it's very different when you know just diving the East Coast versus the West Coast. Just even how quickly our depth drops off here on the West Coast compared to the East Coast, and yeah, there's a lot of it's a great comparison to make. Very
Yeah,
different environments.
yeah, definitely. I had, I
Yeah, and
had,
the causes are so large scale, like currents and topography
Hmm.
of the ocean floor. It's interesting.
And just the fact, you know, that it's the spreading center in the Atlantic Ocean, and it's a younger ocean basin than the Pacific, and yeah, there's a lot of neat contrasts.
I had one opportunity to dive Graves Reef back in 20, might have been early 2010. And I remember it very clearly because we were supposed to be out on the Nancy Foster, and they pulled the ship from us at the last minute because the Deepwater Horizon had started gushing oil from the
Oh,
sea floor, and so we were end up taking day trips out, diving on the live bottom reef every day for a week and a half. But it's a hard sanctuary to try and communicate because it's so far offshore and
mm-hmm.
small, isolated. But it's got a ton of large predators, large fishes that are really extraordinary to dive in and around.
Yeah, it's definitely like an underwater area of just where things converge. But yeah, it's out of sight, out of mind for a lot of people, which is one of the challenges of national marine sanctuaries, especially for places like Gray's Reef or even Cordell Bank, where you know trying to make that connection. And that's where I think you know having the telepresence and the live feeds and being able to sort of figure out ways to connect our communities to these places is really important, and it's helping, I think, to increase understanding and awareness.
Yeah, you can't care about something if you don't know it exists.
Right.
Yeah, that's one of the challenges that my dissertation work focused on, was trying to explain and describe, characterize the deep sea habitats for a lot of the Pacific remote island sanctuaries, or not sanctuaries, but monuments rather, and
Mm-hmm.
some some areas of the Phoenix Islands, which were established as large remote MPAs, you know, some about the size of California, at least in the Phoenix Islands. And prior to 2017, we really had zero knowledge of what was below 50 meters or so. So a lot of this
Mm-hmm.
new data is coming to light, and it's actually pretty extraordinary. We're finding that actually peak coral diversity is around 1,500 to 2,000 meters. So the deeper you go, the
Oh,
more
wow.
corals you find in the bathyal depth range.
Wow, that's that's pretty cool. I mean, that's just shows why we really need to keep focusing on some of these deeper water explorations and characterizing these environments because there's a lot we still don't know.
Yep.
Yeah. Well, and I think, you know,
What
for
kind
Channel
of,
Islands National Marine Sanctuary, it was sort of, you know, up at 2015 was that mapping workshop that they had. It was sort of this revelation of like we've done a lot of nearshore mapping, but for the waters and the habitats within the Channel Islands National Marine Sanctuary, it was over 50% was unmapped at that point, maybe more. And so
mm-hmm.
this working with large ships of opportunity like the Nautilus and trying to really focus on mapping the area and then ground truthing it with the ROVs and characterizing it has really helped us to sort of help to identify where these pockets of deep sea coral community habitats are located.
Yeah, and I think it's something like over 90% of our sanctuary is deeper than diveable depths. And so
Right.
we do a really great job within our kelp forests, our eelgrass beds, and those types of more shallow water habitats. But everything deeper than that, which is the majority of our sanctuary, we need to do a lot of work to better monitor and understand what's there.
Hey Al, can we get a zoom?
It's a nice shot of the jelly.
Yeah.
All right, let's come wide. Thanks.
Well, I know for that trying to communicate, the outcomes of this expedition, we're looking at obviously with the imagery that's being taken, being able to share that with our K-12 community and also our informal science partners and aquariums and museums. And the photo mosaic piece is actually hopefully the outcome from that will be really great to be able to sort of get the dimensionality of some of these deep sea coral community habitats. I know you guys took a couple of, or did a photo mosaic off of, wasn't it, was it Poti Peninsula off the south side of Anacapa, Lizzie?
The sponge, the large sponges.
The glass sponges?
Yep.
Yeah.
Oh, sorry. Yeah, we have some glass sponges. We took a larger, kind of more habitat-like photo mosaic of some big boulder outcrops just southeast of footprint. And then last night, we found kind of it was really tall, and we just tried to circumnavigate the top boulder portion of a big boulder with a bunch of Lophelia on it, which is normally
Nice.
a reef-forming hard coral. But here in the Channel Islands, due to the upwelling events, which reduced the pH quite a lot versus a more stable habitat, such as the Gulf of Mexico, where there's not as much upwelling, they pretty much crumble. They grow and then they crumble. And so they're not really reef
All right, Al, let's
forming,
get another
more
zoom.
than a shell. Ooh, so smooth.
Mm-hmm.
It's a big jelly.
Very fluid motion there. So today, as we go up the wall, we'll look for more opportunities for a photo mosaic. It might be kind of difficult though on a vertical wall, but maybe when we get on some of these shells, we'll be able to come across some really nice habitat.
Yeah,
Look for
I
more
think
photo
so.
mosaic opportunities. Yeah. And I think you were describing on a previous dive, Lizzie, about making some of the 3D models too, which will be great to use for some of our education and outreach. And again, trying to figure out how to engage our communities to increase the awareness and their sort of understanding of these deep sea places and why they're important.
I hope so, yeah. Anything that can bring these really remote and unrelatable systems into the hands of the public and students in a way where they can better interact with them and understand their form and function and the services they provide to the ecosystem and humans, I think would be our ultimate goal.
Mm-hmm.
And we do have some within the sanctuary's ownership. We do have some skeletons of Lophelia, but they're so delicate, and we only have like 1 or 2 specimens. They're requested a lot by the other sanctuaries for public education and outreach events. So these 3D models, which will be much more, oh, look at that, we got some black corals on this rock here to the left. Bathypathes, probably.
Beautiful. Yeah, the models will, I think, yeah, it'll just be better for outreach purposes.
Oh, Meredith is saying alternopathes. Thanks, Meredith.
Oh, well, I was going to go for the corals, but this jelly decided to... All right, Al, let's
They
zoom
still
in.
want some attention. Kind of got it all going on there. We've got the jelly, the cucumber, some stars,
Actually,
brittle stars.
Couple sponges.
polyps look really weird.
Oh,
Yeah, let's
Very
zoom.
opaque.
Let's zoom further in now.
Got it.
So Meredith's saying is it used to be Bappy Pappies. They changed the name to Alternatipathies.
Alternatipathies, yeah.
Alternative, yep, you got it.
All right,
Because it's
better
more
come
PC.
wide.
Very cool. So this is an example of how corals typically prefer hard substrate. So you will not typically find them going in the sediment unless they're sea pins. Oops.
Bridge nav.
We've got some thorny heads joining our
30
within our
meters,
view.
bearing 036.
Passed over a few of them.
Yeah, it looks like we might have a couple different species of sea cucumber that we're seeing. We have some different morphologies anyway.
All right, Al, let's go for a zoom on this small jelly.
Nicely done. It's traveling quickly. Oh, so cool.
Oh, and it went away.
Yeah.
Steve, I think Mike Brennan had written something up about those jellies. I don't know if he ever published it.
Really?
Yeah,
Oh.
informally known as the scrubbing bubble jellies. It was
The genus name is Benthodon. I didn't know that.
yeah.
Scrubbing bubble
Mike
jellies.
Brennan?
I like that.
Yeah, it was a weird one for him.
Huh.
But
Yeah.
we had seen so many on a cruise in 2017, maybe, that he decided to write something short up.
We're also seeing every now and then some drift seaweed. So we're seeing some seagrass and some kelp pieces on this slope here.
Oh, you know what? They were pedunculata. Is it
I'm not
different?
familiar with it.
Yeah, they look very similar, but different. Different genus, I think. And it was just a short article. I don't think it ever got published. We saw them in really high densities up in, I want to say Greater Farallones National Marine Sanctuary, but it's 2016 or 2017. And I think they have shorter tentacles, but similar shaped bell.
More sea cucumbers, and every now and then you'll also see these little orange shrimp-like things floating by, which are mysids.
And what are mysids, Lizzie?
Mysids are a shrimp-like creature. They're a crustacean, but they're their own group. So they're slightly different from a lot of the other like decapod or the shrimp and crab species that we associate with crustaceans. And I believe it's one of the characteristics that sets them apart is that they brood live larvae. They don't lay eggs. Nice.
Oh, that's beautiful.
Wow.
Oh.
What species is this?
Oh, and it's gone.
Nice shot.
That was a beautiful shot.
Yeah, jellies are nice because they fly up near the camera and then don't move very fast, so.
I love the symmetry
Yeah,
of
go
them.
for it.
Mm-hmm.
All right, we got the same number. Makes me happy.
It's defaulted to 3.
Oh, just didn't want to change it?
I didn't change it.
I was going to say, that's probably a 3, but I'm not sure.
I think that's an eel pout we just passed center screen.
Yep.
They've got big heads and short little pectoral fins that are very close to their head, and these elongated bodies and dorsal fins. Eel pout.
I would imagine the thorny heads and eel pouts are feeding on some of the benthic infaunal organisms, worms, crustaceans.
Infaunal and maybe some epifaunal, just right on the surface.
Mm-hmm. I mean, there's not much else.
Yeah.
This is it.
Maybe some smaller mysid species or other like isopod, arthropod. I wonder if they eat snails. Tom was saying the rockfish tend to be opportunistic, and so I wonder if they eat like small snails too.
Probably. Get hungry enough.
And we also get those like skeleton shrimp down here as well, which we can see sometimes in kelp forests. They're kind of like the elongated, segmented shrimp with almost little praying mantis arms.
Mm-hmm.
These little shrimp?
Oh no, they would be on the sediment that we can't see.
Oh, got it.
Yeah. Almost like the caprellids.
Kelp holdfast. Little one.
There we go. Is that what I'm trying to say? Caprellid?
Yeah, the little. They get all over your wetsuit in the shallow depths sometimes.
Yes.
Especially during West Coast observations when we're grabbing buoys.
Oh, I missed that.
Well, and you can see, I mean, so the thorny heads they're very low energy. I mean, they're conserving energy. They sort of wait until the opportune time. They're not going to do a lot of swimming around, searching.
No. If they're not swimming away because of the ROV, they're not going to swim much in general, I feel like.
No, I don't feel like they would either. I think they just conserve energy.
And we're only at 3 micromolar oxygen, so that's valuable commodity here.
That's true. Good point.
And another eel pout lower
Stone
left.
rock.
Whereas last night when we were, in the shallower depths off of San Miguel Island and all the rockfish, and they were moving around and much more active.
Hmm.
Ooh, is that a sole?
Oh, nice flatfish there.
Yeah.
If they touch, is that a puddle?
Different dive, different dive.
Different type. Okay.
Looks like a couple species here.
Yeah.
All right, let's get a zoom.
Got a small mouth.
Is
That's
that a
like a
Dover?
turbot. Or maybe a
Still can't tell them
that
apart,
one looks
honestly.
similar. It's got the yeah. That they look like they're similar, huh? They have the same pattern on their dorsal ridges.
Flatfish are really cool because when they're larvae, they start out like a normal fish where they have two eyes on either side. And then as they settle and mature, they turn over to one side and their eye migrates from what would be the bottom side up to the top. And this all happens at a pretty young, early stage before they develop their coloring, and whichever side they settle on becomes the white side, and whichever side is facing up becomes the pigmented side that we just saw.
So it
Erica
varies.
said that was a Dover.
It varies on what side.
Dover?
Yeah, there's definitely more common sides, and then every now and then it'll be a little more rare to get a different side, but there are like right versus left in ways that they settle.
Right. Also, well, it's hard to see with these images, but another key distinguishing characteristic is the lateral line, and whether if it has a hump to it or not is a way to identify.
Mm-hmm. What is a lateral line, Julie?
It's a sensory organ along the fish that helps them detect pressure changes in the water, and it helps them to be
All
aware
right,
of their
let's get a zoom.
surroundings.
Yeah, that's how fish school so well, and they're able to keep equal distances from them and their neighbors.
Trying to look at the difference between a Dover and a rock sole.
Cool, let's come
I
live.
noticed those little the white sort of splotchiness on the margins of the dorsal area where the fins are, and the rock sole has those. With Dover, not so much.
How far are we from BF, roughly?
I was just going to mention that. We are about 280 meters off of BF.
Okay. Since we're moving that way and we've got that much time, should we do a transect upslope?
Sure can.
Alrighty.
We can start anytime.
Yeah.
All right. Let's do it.
036.
036. Well, Science, is this bearing look good for you on HIPAC?
Yep, this looks great. We're kind of following this almost ridgeline contour upslope.
Alrighty. Let me know, Paul.
Just gonna get set up here. Just gonna get started so we get out of this cloud a little bit.
That works. When the cloud is passed, we can start transect.
Anytime.
Roger. Let's. Now seems good. Bridge nav. 200 meters, bearing 036. Transect 1, starting now.
Thank you kindly.
Anytime.
Oh, and Daniel was pointing out that we came more across of those snails that we had zoomed in on in the beginning of the transect, the Bathybombix. We saw at least a few of them, so we'll continue to try to keep an eye out for those.
I bet he will.
So we are conducting transects to more, to collect data in a more standardized way. And so we can compare the habitats we're encountering here to other spots on the West Coast, because we're trying to maintain a fixed heading in the direction that we're going, a fixed altitude above the seafloor, so we can estimate the distance and the area that we're covering during these transects, which will help us later calculate the density of some of the organisms we're seeing, like these thorny heads, and if we encounter any corals or sponges. It helps us characterize the environment in a non-biased way, where if we were kind of exploring and meandering, we would be trying to chase down all the cool things versus keeping a fixed heading, we're more randomly sampling the environment as we encounter it, so we can definitively say more about the things that we're seeing. So we can compare between sanctuaries, because we've been doing similar transects in like Olympic Coast, for example. We can compare inside of sanctuaries to outside of sanctuaries, see if there's any differences in the habitats we encounter, or if there are similar habitats, how are the organisms potentially different within those habitats depending on where those sanctuaries are. And then at the same time that we're doing these transects, we're also collecting environmental information such as the temperature, the oxygen, the dissolved oxygen at these depths. We've got salinity, which helps us understand the differences between all these transects that we're doing, and help explain why we're seeing certain things and maybe not others.
Mm-hmm. Yeah, I thought the comment earlier about the percent oxygen in some of these areas and how that's a variable in different environments, and that will impact, the types of organisms that will live there and their behaviors.
Mm-hmm. And what we're really doing is collecting a snapshot. So these fish will move, the currents will change, and then there's something called the oxygen minimum zone, which you might have heard us reference before, which is just a depth or an area,
We can just stop and
a
pause.
water layer. No worries. I can mark down that time.
Yeah, Science, do you mind if we pause here to investigate our potential wrap in the tether?
Yeah,
Sure.
yeah, absolutely.
Bridge
No
nav.
worries at all.
So if you can look up, I think that's channel 2 shows the
Hold
Argus
position.
view of Hercules, and right now it looks like we've got a little bit of a twist on the tether. that
Yeah, we
need
just
to
want to make sure that
pause our
that's,
transect.
yeah, we'll make sure that's out before we keep moving. And I'll just mark here that we paused.
And that is important because that is the main communication between the ship to the different vehicles, and also the power sources. And so you don't want to have kinks in your tether. So Sam, is that a matter of just waiting it out, or the ship moving its position a little bit?
It'll be actually it's looking a lot better now. It's probably going to work itself out, but it's usually a combination of the Hercules pilot just maneuvering the vehicle either clockwise or counterclockwise to take out that wrap. But now that we're looking at it, it might
It might undo
not need
itself.
that. Yeah, it might undo itself.
Okay. So these transects that we're doing in the deep sea environment with ROVs, we do these. This is a methodology that's used to characterize all sorts of systems, terrestrial and marine. It's a standard survey technique that scientists use.
Yes, good point. You can fly a survey. You can use the ROV for a survey. You can scuba dive and swim a transect survey.
Yeah.
Many different
Sometimes
ways.
we'll use, if we don't have instrumentation like we have on the Nautilus, we can use transect tapes and compasses, and yeah, it's a systematic way of surveying and characterizing an area in an unbiased way.
Yeah, I used to do transects in the intertidal zone.
Me too. In fact, we do that with our citizen science youth program, Limpets, which is a long-term monitoring program and experiential training for students. And we have vertical transects on some rocky intertidal benches, and in other ones we do random plots off of a transect, which you just generate random numbers, and that tells you where you put your plots off the main line, either to the east or to the west. And it's all about sort of just, again, not going to those hot spots where everything's really great, but just taking a snapshot of what's there.
Is Limpets still going on despite what's with the pandemic this year? Or are they on
We
pause?
haven't been able to. Yeah, it's been tough trying to get students into the field this season, and right now we're doing online learning with teachers and still using the curriculum and teaching students about how they would, what the scientific methods are and the protocols that we use. But yeah, the actual field monitoring component is on standby until we can actually get into the field in a safe way with COVID.
And I mean, that makes totally reasonable sense.
Yeah, I do know the coordinators.
Like
We've been going out and doing some baseline monitoring of certain key locations just to keep the data, the baseline data going.
Yeah, citizen science is a really great way to incorporate the public and gather information that we wouldn't be able to just by ourselves.
Yes. Well, monitoring is expensive, and and as you can see, I mean, we're hitting one little spot at a time, but if you can do a little crowdsourcing, get some citizen science initiatives going, it's not that to say that you would replicate exactly what's happening when the experts go out in the field, because I mean, some of the taxonomy for being able to identify species, it takes years of training and experience, but there are some species that the public that are they're really conspicuous and easy to identify, that the public can help gather information on those organisms.
Mm-hmm. Yeah, and some monitoring really is just as simple as counting things in a square.
Mm-hmm.
And so that's still incredibly useful information that we need. And so it's kind of like volunteering for a park or for any other organization that you like or you believe in, or if you really enjoy nature and being outside, this is a great way to do it while also contributing to some other bigger objective.
That's true. I mean, we have volunteers that help us with whale identification and using different apps, the Whale Spotter Pro or Whale ID apps, and then we've got birders that go out and volunteer on the beaches to help do inventory of shorebirds and seabirds offshore. There's a lot of different ways that you can contribute to our understanding of the pulse of what's going on in a system.
Mm-hmm.
That looks better.
Good to go? Alrighty. Yeah. So science, we didn't come too far off of our where we paused, so okay to continue ahead?
Yeah, that's fine.
And would you like this to go a little farther then, past our original 200 meters, just to make up for the couple of meters?
Sure, why not?
Okay.
Yeah, we can go a little bit farther, but starting from here is just fine.
Roger. Bridge, Nav.
Thanks.
.150 meters, bearing 036.
A little rock in the upper right corner.
Yep.
Hoping to see more of those soon.
There goes another eel pout.
Those are so cute. Love a good eel pout. Yeah, so the beginning of the dive, I definitely anticipated more soft sediment. So we may not get in our shift up to the part where I'm expecting hard substrate, but I'm hoping maybe between BF and BG and BG and BG1, those different waypoints that we have, that we'll be seeing some rockier substrate. So we'll see. I feel like we lucked out last night, Julie, because the beginning of last night's dive kind of started similarly, where the first third was really mostly soft habitat, and then when they switched watches, the following watch, you got all of the rocky substrate, and then we got to finish out with the rocky substrate too.
That's true. Sometimes you gotta take one for the team.
Mm-hmm. Definitely means I'll be tuning in throughout the day, though, while trying to catch up on some sleep.
Yes, me too.
I did that the other night. There was the last dive on the Santa Lucia Bank. It's roughly the right time of year to see some of these flatfish, the petrale sole. They do these breeding aggregations, which we really have not, for a lot of these fish species, seen or witnessed much breeding behavior. So a lot of that reproductive behavior, it has not really been super well documented. It is for some species, and for others, it's just a black box. And I forget how many years ago there was a there's this little black fish in the middle of the screen that I've seen a few times before, and I have no idea what it is.
I've definitely also seen them before, and it's an interesting fish, and I can't remember the name.
So small.
It's not a poacher, is it? It kind of reminds me if I was shallower. I don't know. I'm gonna look at my book.
It could be. Deep sea poachers. Yeah, so the last night of Santa Lucia, it was a short dive because the weather was coming in, but again, we've been partnering with a lot of different experts and partners across NOAA and outside of NOAA to stand these watches, and we've been partnering with a lot of National Marine Fisheries Service folks. And they had noticed on a previous visual survey they had done with an AUV, or an autonomous underwater vehicle. They had gathered a lot of photos around this time of year on Santa Lucia Bank of a petrale sole breeding ground or behavior. There was a bunch of them. They were all piled on top of one another, with the biggest flatfish on the bottom and the smaller flatfishes on top. And they were interested, since it's the same time of year, if we could go back and get some ROV footage of the same kind of aggregation. And they started, I think they started at 8 p.m. and went to 4 a.m. So it was a pretty short dive, and I had just had a shift or something. So pretty much the National Marine Fisheries, our coworkers, colleagues, stood watch for those shifts. And I had my computer on my bed playing as I was trying to fall asleep, and then every now and then I'd wake up and it'd be like, oh, are there any fish? And then I would go back to sleep. So I really didn't get any restful sleep for until well after midnight. So I was like, well, I guess I shouldn't do that anymore because I'm exhausted.
Because then you're ruined for a little while.
Oh gosh, yeah. Tom Leidig, one of those National Marine Fisheries Service colleagues of ours, just popped on and said that little black fish looked like a small slickhead. There's another one there on the, that just exited bottom left of our screen.
What's a slickhead related to? Let me look that up.
Great question.
The hipster fish.
All the really dark colored fish intrigue me a lot because I am just imagining it to be one of those really interesting looking deep sea fish with either lots of teeth or really big eyes, or bioluminescence. But those are so small that it's hard to tell from the ROV footage.
So if we can get a close up, they are characterized by an unscaled slick head. That's why they have the name. The skin is slippery and the flesh is flabby.
All right.
It's not a great way to be described.
Okay. Dude.
Some have no teeth.
Yeah. Oh.
Others have fang-like teeth. And they could have light organs too, so that could make them bioluminescent.
Oh, cool.
But most are blackish. Yeah. This is really cool. I've never seen a slickhead. Thank you, Tom.
Yeah, Tom's saying it's so hard to get a mid-water close-up, or you can't really ID them until you get a close-up, which is also difficult, in particular for the species, because you might have noticed they're just kind of zipping around the screen and kind of skittish.
Yeah. Well, that goes on my, it wasn't on my life list, but I'm going to put it there. A slickhead, something new.
Yeah.
Are there other deep sea organisms on your life list?
No, I mean, I don't know. There could be.
Oh, the barrel eye.
I mean,
I've always
this
wanted
is
to see a
totally
barrel eye.
totally new for me, right? So
Okay.
it's, you never know. I'm open.
That's fair. I agree on the barrel eye though. Those are.
Oh, have you guys seen any throughout your various missions?
Not this year.
No, I feel like every now and then we see one, but we've never had a good enough view of one for video.
Well, and I guess you don't really hang out in the midwater much, which I'm assuming is where you'd be more likely to see them.
Yeah, and I feel like we've seen one on ascent or descent, and so it was like a blur.
Mm-hmm.
That would normally be a video highlight 5. Well, maybe not for
Only a
Steve,
5?
but oh, it's out of 5 out of 5.
Oh! I thought it was out of 10. No wonder I thought Steve thought everything was lame.
Steve's still hard to impress,
but
it's a relative scale.
Yeah.
And it's very subjective. And I think we need a protocol to describe what the different criteria are.
It's actually
Julie,
something
Tom's saying a hatchet fish should also be on your list.
Ooh.
Oh,
hatchet fish.
Yes.
It's like a 3.
We did see hatchet fish the other night, actually.
I have seen hatchet fish. Those are they're in the mesopelagic layer, right? I've caught one before in a bongo net, not on an ROV though.
Yeah, that sounds amazing. We saw one had just come up and was swimming right in front of the Herc's camera lens. So we got like 3 seconds of it just like fluttering around and then disappearing. But that was so cool.
They're pretty tiny.
Yeah, they're very small. I mean, most midwater fish you see are actually really small. It's just the photos and all the informational books are like blown up and really cool. And then if you're ever encountering them in the field, they're so small.
70 meters left on this transect.
Thank you.
Yeah, we used to run this bioluminescence night cruise off of Dana Point back in my former job. And so we would go out during a new moon and we would do some trawls and we put a squid light out and we would get market squid and also the, what's the bigger squid? I just totally slipped my mind, but it'll come back to me. And then when we do these midwater tows, we would get different just the hatchet fish and some of the lanternfish, and it was really neat to see how different it is at night than during the day. And I know that whole layer sort of does a vertical migration up near the surface and then goes back down. So it's some really cool adaptations. And just sort of highlights how different communities use the ocean in different parts of the day.
Yeah. I think Tom said that was a snakehead eel pout.
Snakehead eel pout.
Mm-hmm. I knew it was an eel pout, but not sure if it was a particular.
Well, it was quick. I'm impressed, Tom.
Yeah. Yeah, it was a snakehead. Cool. Sea cucumber is about to tumble down this slope. Yeah, and we're seeing more of these snails. Daniel is saying it's in the middle right part of the screen. If there's any with anemones on them, it could be also a potential sample interest.
Yeah.
So the slope is getting steeper.
Excellent. We are expecting that at some point. Now is as good a time as ever. Oh yeah, so if we had like zoomed in, sometimes you'll be able to tell. It just depends on the orientation of the snakehead eel pout. But if they're kind of angled at you in the right way, they're easy to tell apart because they have iridescent reflective scales on their pectoral fins and their lower jaw. So you'll just see the little shiny surface that's just on the lower back portion of their head and their pectoral fins.
Well, they're going to have to slow down so I can check that out.
Yeah, yeah. Once we stop transect, we can definitely take a zoom. They're really cute. If they don't flutter away before.
Well, I wasn't talking about the ROV. I mean, they're darting off. They need to stick around so we can look at them.
Yeah. More thorny head. As we're nearing the end of this transect, as you heard, we're getting steeper, which is a part of the plan. We're hoping to get so steep that there's just no sediment sticking to the wall of this canyon, and we'll have some hard sediment, or excuse me, hard rocky substrate that's exposed where we hope corals and sponges might be growing. We want to count them.
Yes, we do.
Seeing lots of these snails that Daniel had pointed out earlier, so there's probably a good chance we might pick one up. Yeah, Tom is saying these are long spine thorny head, and you can tell the difference between this species and the species called short spine thorny head by the height difference of their third spine on their dorsal fin.
Dorsal spine.
So it'll be longer, longer than the first two, and a short spine it'll be the same size, if not shorter, than the first two. But a nice
and do we expect
way to
and
tell
it could
them
be
apart.
mixed?
Well, yeah, that's what I was saying. So it's nice because the long spine thorny head tend to be much deeper, and so we're probably not going to encounter any short spines in this portion of the dive, at least. They segregate they have these niche habitats.
Oh, you can see the long spine there. That's the pectoral fin.
Yeah.
I'll go another look at it when we go by. It's quick.
Oh, knocked over that sea cucumber.
It's like Chevy Chase Vacation. Look while you can see it quickly. Gotta move on.
So we are at 20 meters to the end of this transect.
Okay.
Awesome. Yeah, next time we see a couple of these snails, if we could get 3 of them and probably a slurp, we can send that off to Daniel. It's interesting some of these sea cucumbers are kind of making this L shape with their mouth parts up into the current.
I was noticing that too.
Yeah, it's kind of strange. Not all of them, but there have been a handful of them that have been doing that. Tom says they're waving at Herc.
That's what it is.
Oh. More drift algae.
There's a flatfish there.
You could probably use that to tune in the PID, you know? That would be an interesting experiment.